Hello everyone, this is Xing Jianzhen from University of Wisconsin-Madison. Here I will talk about our new project, Outer Structural Connectivity Detected with Dilated CNN Analysis. In this presentation, I will cover the background, the processing of the data, our method, and our findings. In this study, we want to find the macrostructural difference between different groups. The data we use is the DTI imaging, where each voxel is a 3x3 symmetric positive definite matrix. From the raw DTI, we use the tractometry to get the fiber bundles, which is the object we care about in this project. To get the fiber bundles, we use the preprocessing steps called TractSeg. We first register all the subjects from subject space to template space. Then apply TractSeg to get the masks for each fiber, then the starting and ending points of them. After this, we use the tracking method to track the fiber bundles from the starting points to the ending points. This is an example of the FA image in subject space. This is the mask of the CG segmentation after TractSeg. Finally, these are the tracks of CG. To train a model on the fiber bundle, which is a 1D sequential data, we apply the dilated CNN model. This model, compared to the traditional CNN model, includes holes inside each filter. So the view field expands exponentially as the layer goes deeper. We train the model so that we can predict the last voxels from all other given voxels. If the model can predict well, we can claim the model is a representation of all the input data. We first train two models from the positive and negative groups and compute the original distance between two models as the representation of the difference between two groups. Then we shuffle the data and train another two models, and we shuffle again. By permutation test, we can have the distribution of the distance between models. If the original distance lies in the extreme side, we say that the fiber is statistically different between these two groups. We apply our method on two datasets, the ADRC, which uses PIP positive and PIP negative as the biomarker, and DIAN dataset, which cares about the gene mutation carriers versus non-carriers, to study autosomal dominant AD. All participants are cognitively healthy. These are the fibers we found different. Between two datasets, we found 9 fibers in common and total of 21 fibers out of 50 to be impacted by AD in very early stage. Further studies are needed to determine the temporal relationship between the accumulation of amyloid and neurodegeneration in the development of dementia.